Hello everyone, welcome to our Ant Biology webinar series. Today we are very much delighted to have Professor Arun Bandyopadhyay as our speaker. To all our audience, you can post your question in the comment section below. Continue till the end to know much about the work the speaker is going to say. Hope you will enjoy the talk. Feedback links will be given at the end of the talk, so stay tuned. Now, without further delay, I would request our mentor and convener, Professor N.R.I. Banerjee, to welcome Professor Arun Bandyopadhyay with our audience. Over to you, ma'am.
Thank you, Shahli. A very warm welcome to you, Orunda. Although I cannot see you, but viewers tonight, and doubtless to the future viewers who will be logging in to our platform where Professor Bandyopadhyay's talk will be live. A few words about ant biology, translational outcomes research group, Department of Zoology, University of Calcutta
Your
is
voice
delighted.
has a problem. We cannot hear you.
I think there is some low connection of network,
Yeah.
so please wait. I think there is some network connection problem of ma'am there.
Yeah.
That's why she has left.
Sorry,
Yes, we can.
I'm so sorry, I got disconnected. So I'll start from the beginning. I do not know how much I was hurt. This is something that none of us have any control over, so I apologize. A few words of introduction about ant biology, which Translational Outcomes Research Group, Department of Zoology, University of Calcutta is delighted to present. As the ampersand in ant biology indicates, biology itself as a discipline is multifaceted. And I think today's speaker will elucidate that point all the better because he is the first speaker to have been invited the second time to our webinar. Earlier, he spoke on our progress and prospects in biology, where he talked about nurturing creative mentality. And today, he will be talking about the core science. And this is exactly what ant biology wants to embody in the sense that we try to bring to you scientists working in the core sector of biology and academics and professionals with expertise in other domains of biology, looking at biology from a slightly different perspective. Today, our speaker, who will be introduced by our moderator, virtually needs no introduction, and a lot of your students, Professor Bandyopadhyay, are eagerly waiting to listen to your talk. Ever since your last talk, there have been a lot of requests that we want to hear about your core science, and you are so strong. And despite your busy schedule, you agreed to speak tonight. So, without further ado, again, a very warm welcome back. our webinar. And Shahili, may I request you to please introduce our speaker today? And we are all waiting to hear your talk.
Okay, ma'am. Now I'm going to introduce Professor Arun Bandyopadhyay. Professor Arun Bandyopadhyay is the director of CSIR Indian Institute of Chemical Biology, IICB, Kolkata. His research interest lies in the identification of protein biomarker for risk assessment of cardiovascular disease, understanding biochemical basis of compromised reverse cholesterol transport in humans, inflammation resolution in plaque stability, and implication in atherosclerosis, unveiling mitochondrial dynamics in cardiac hypertrophy, miRNA mediated molecular regulation of mitochondrial dysfunction in cardiac hypertrophy, and identification of small molecules for the management of respiratory disease. He has received several awards and honors. He has a number of patents and almost 50 research articles in his name. Now, sir, I'm requesting you to start today's talk, and over to you, sir.
Okay, thank you. Thanks, Riley, for nice introduction. Thanks to Professor Ena Banerjee and Department of Zoology for giving me this opportunity to speak in this seminar series. Yes, it was a very nice interaction in last seminar. It was a very popular talk. I spoke on that issue in several audience, several occasions, but at that time it was not a technical presentation. So today I am going to present really my technical presentation, what we do in our laboratory. So let me just share it is already shared. So slides are visible now,
Yes, sir.
Sally?
These are visible.
Yeah, okay.
Yes, sir.
So basically we work in cardiovascular disease. So the topic of this today's presentation is cardiac complications, unresolved inflammation due to dyslipidemia. My main interest is to understand the inflammatory process and the dyslipidemia in the progression of atherosclerosis plaque, which ultimately leads to cardiac failure. So the coronary artery disease is all known. It's a set of conditions of jeopardized myocardial blood flow. The clinical manifestations associated with acute CAD or coronary artery disease includes ST segment elevation myocardial infarction, non-ST segment elevation myocardial infarction or STEMI, or unstable angina. So basically, the cardiac failure or cardiac heart attack can be diagnosed by ECG. So in ECG, there is a ST elevation. In most of the cases, the ST elevation will be there, and this is called STEMI, or in this ST segment elevation myocardial infarction. In many cases, there will be no ST elevation, but there will be ST depression, that is called non-ST segment elevation myocardial infarction. So the samples which we use from the hospitals, the clinical samples, I use STEMI. So henceforth, I will term as the MI. So so let me introduce the topic first. Basically, the cholesterol is a main culprit, but cholesterol is required. Cholesterol is synthesized from liver, and it is transported into the peripheral tissues for function. It is necessary for the cells to function. But excess cholesterol is returned back to liver via a process called reverse cholesterol transport with the help of HDL and macrophage and the transporter proteins, and it is the recycled, and it is actually degraded in hepatocytes inside the liver. So that's the way cholesterol level is maintained, but it is not always the case. So the excess cholesterol, or the cholesterol which is modified, or the low density lipoprotein is modified because of oxidation or the oxidative stress. It is modified into LDL, modified LDL, and the modified LDL is engulfed by the macrophage. Then macrophage is converted into foam cells, but the excess cholesterol, as I said, that it actually recycled back to liver with the help of HDL and transporter. So as long as this process is maintained, and as long as inflammatory mediators are in the low concentration, and it is the balance is towards the pro-resolving mediators, the plaque will be stable. Stable atherosclerotic plaque will be developed. A small necrotic plaque area will be there. So this is called the resolved plaque. So most of the people will have the plaque with the aging, with the normal aging process, but that plaque remains stable. So as long as the plaque remains stable, there is no issue. But when there is a problem, the pro-inflammatory mediators on the upper side, the pro-resolving mediator in the lower side, the balance is shifted towards pro-inflammatory mediators. So inflammasomes, along with the other molecules, it is shifted towards chronic inflammatory stage, and because of chronic inflammation in the intima layer of the aorta, the large necrotic core area, lipid or core area is developed, plaque is developed, plaque become unstable, and then there is a thrombus formation because of the coronary the heart attack. So this is called coronary, and the coronary occlusion will be there. It's called heart attack. So this is basically the process. So there are two main players which we are interested here. One is the cholesterol transport, another the inflammatory process. So henceforth, in the 30-minute talk, you will be listening to reverse cholesterol transport and the inflammatory process. So what was our question? So we asked the question that whether these molecules will be the alteration will be reflected in the blood. Obviously, blood reflects everything. So that's why we thought that let's check the blood profile and what is there in the blood. So but our approach is to collect samples from the patient samples, patient in the admitted in the Apollo clinical hospitals. So patients entering into the hospitals within 6 hours of chest pain, presumably within six hours, maybe more, then sample is collected by the doctors, and then it is transported into in the laboratory. It is processed, and the proteins are then analyzed by using high resolution mass spectrometry or the orbital mass spectrometer, and then we try to analyze what are the proteins altered in the patient samples, and also we compare with the healthy control samples. So basically, this is mostly done by these two students. Babita Andas finished PhD, and Komalika was a postdoc in my lab for some time. So as when we so the process is to do the proteomic study, but at the same time this plasma sample is very valuable sample. So we try to understand and try to find out some molecules, some proteins, or the candidates based on our hypothesis. So since at the time of coronary artery disease or acute coronary syndrome or heart attack, there are a lot of process changes inside the blood. So we identified a protein based on our hypothesis that's called STLT1. STLT1 is a soluble shame-like receptor protein. In any CAD subjects, CAD has a high thrombin level in the plasma. Thrombin is protein platelet activators. So STLT1 is released from the platelets in response to thrombin stimulation, and STLT1 is directly related to chronic macrophage-mediated inflammation. So you can see there's very interesting data we obtained from human samples. The control, the STL level is very low, and in the patient samples, patients who are having heart attack, the level of STL1 in the plasma detected by ELISA was very high, significantly several folds higher than the healthy control, and it is reduced significantly when after the coronary artery when it is corrected by CABG or the other process. But there is an interesting story here now. See, the amount of protein, the STLT1, is increased very remarkably in the patient samples. But there is a group called asymptomatic group. In asymptomatic means patients with having the risk factors for atherosclerosis, like high lipid profile, abnormal high blood pressure, and other risk factors, smokers. So in these groups, the protein SCL1 level is very high, significantly high. So it shows that it can be a predictive marker for ACS. Much before the heart attack happens, this protein level is high. So along with many other experiments, I just summarize this part only, that we showed that STLT1 binds to a protein called Fc gamma R1. We identified this protein through bioinformatics analysis, computational biology, and the wet lab experiments in the macrophage. And we have found that this protein, after binding with Fc gamma receptor, STLT1 elevates the BTK activation, causes BTK activation, MEG activation, and then through this pathway ultimately activates the NF kappa beta, so inflammation induces inflammation in the macrophage, and the secretion of TNF alpha and maintenance of inflammation. So the protein which is released from the platelets at the time of heart attack or before heart attack helps in the maintenance of inflammation, or it drives the system towards the chronic inflammatory stage. So this was published last in 2019, late 2019 December, as in the Clinical Science as a cover page article. So if you open this paper in the journal in Clinical Science 2019 issue to December issue, November issue, you'll find the cover page is from our paper, our data. So now coming back to the orbital analysis. So we have this is the study design. So we pulled 9 control samples, 9 blood samples of 9 healthy control subjects, and also we pulled separately the STEMI cases like the myocardial infarct samples, and then we subjected to orbital analysis as well as SWOT analysis. So the SWOT is a different type of mass spec analysis. I will come to that. And then, so basically both are proteomic analysis. So proteomic analysis of plasma samples by Orbitrap and LCMS and SWOT LSMS were conducted. The results were analyzed by pathway annotation of identified proteins using IPA. This is a very strong software for analysis of the pathway. And the differential express proteins are identified. These proteins are shortlisted, data mining, and then finally these proteins are validated. So there are 2 stages in the proteomic study. One is the discovery phase. This is the discovery stage, and this is the validation phase. So let's see the discovery phase. At the same time, we collect data, the demographic parameters from the samples, from the normal healthy control as well as the patient samples. We like age, sex, total cholesterol level, the HDL level, LDL cholesterol level, triglyceride, SBP, the blood pressures, and the smoking status. You can see in most in these STEMI subjects or the MI subject, most of the parameters were higher, like cholesterol, but HDL was lower, LDL was higher. So this is a dysregulated cholesterol or dyslipidemia, in spite of they having the medication under medication, because these people are having SCS. Mostly they know and they knew it, and they are under medication. In spite of that, cholesterol level was abnormal, but sometimes it was the blood pressure was under control. So this is these are the demographic parameters, very useful for analyzing the data in the later phases. So mass spectrometry data revealed that a total of 2,428 proteins are identified in STEMI in the MI, and a total of 2,654 proteins are identified in control. And then out of that, 423 proteins are found to be common in both the groups, which are repetitively unrelated proteins. This is the fold changes of identified proteins. These are displayed by volcano plot analysis by sieve. So using sieve, we can find out the differential express protein, but this is not a real differential expression. This is basically the relative quantification, and based on that, we found that these are the proteins which are altered. The differential express proteins identify Orbitrap and as measured by sieve 2.2. Using sieve 2.2 software, we can find out what are the proteins which are upregulated, what are the proteins which are downregulated. So some of the proteins we identified, those are differentially expressed, are cytoplasmic actin 2, PGLO-RP2, ABCA5, ZZP1, and some of the proteins involved in the complement cascade. I am telling these proteins because these are the proteins which we use for validation and understanding the pathophysiology of coronary artery disease. I'll come very soon. So meanwhile, we have also conducted the SWOT MS analysis. SWOT is actually acronym of sequential window acquisition of all theoretical mass spectra. So using this, we can quantify the proteins. So these were analyzed in our CSR NCL Pune laboratory, and this is the heat map analysis. The total 64 proteins were identified by SWOT MS, 26 proteins were downregulated, 38 proteins were upregulated. Out of these 64 proteins, 26 proteins are downregulated and 38 proteins are upregulated. This is actually a differential expression protein. This is the direct quantification using SWOT MSMS. So this SWOT MSMS can be useful for the quantification of the proteins, and we can find out the differential expressed protein. So let's see what are the proteins that are differentially expressed. So in using Orbitray, we have found C17 or F57 are downregulated. ABCA5 was upregulated. This is very critical. ABCA5 is a protein which we have actually selected for further study. And some of the proteins using in the mass SWOT MSMS based proteomic study, we have found calicin is downregulated, PGLRP2 is upregulated, and AZGP1 was downregulated. Using the signal analysis using various software like IPA. So based on IPA, proteins are subgrouped into two categories: transporters, inflammatory, and coagulatory proteins. 6 previously unreported proteins are chosen for further biochemical validation. These 6 proteins are either related to RCT or development of plaque morphology. So this is very important issue here for the students. See, there are a lot of proteins are altered using mass spectrometry, or if you do the genomic study. But it depends on your thought process, your wisdom, and your target. What are the proteins which you will select for your further study? So you have some hypothesis. Based on that, you select what are the proteins you will select, choose for further study. Also, it is dependent on the reagents and the chemicals available. Based on that, we select some of the proteins. So with that, the philosophy, we selected some of the proteins which are further validated using the Western blot analysis, Western blot, and sometimes ELISA. So you can see C17 or F57 was actually downregulated in the mass spec, and we have validated, yes, this is downregulated. ABCA5 was upregulated. Yes, we have validated using Western blot. The ABCA5 was upregulated, and we have samples before coronary artery angiogram, angioplasty, and after angioplasty. So post PTCA means after angioplasty. So after angioplasty, the before angioplasty, after that time of heart attack, ABCA5 was upregulated, and after post PTCA angioplasty, ABCA5 was somewhat restored or downregulated again. AZGP1 was also very similar. This is a very novel adipokine, AZGP1. So we studied further using this protein. PPR gamma was upregulated. This is involved in this inflammatory pathway. PGLRP2, calicin, and calicin also altered. We validated as we have found in the Orbital analysis. Now let's see AZGP1. What is AZGP1? AZGP1 is a candidate protein for further study. What is it? AZGP1 was found to be novel adipokine decreased in STEMI. It's an adipokine. It is secreted from adipocytes, and it was not. Nobody has reported this protein using the mass spec analysis. We have first time shown that this protein is altered in the during a heart attack. It was found to be connected with most of the candidate proteins using bioinformatics analysis. I'm not showing all this data. So it was selected for further downstream study at the cellular level. We are using computational biology using this software, Class4 2.2, PathDoc Beta, and Haddock. We found that CD36 is the candidate protein which binds with AZGP1. So AZGP1 interacts with CD36 and further confirmed by isothermal titration calorimetry, which confirms the interaction of AZGP1 with CD36, which is further validated using the IPA, immunoprecipitation analysis. So this is the primal view of most energetically favorable model of interacting AZGP1 and CD36, showing interacting surfaces in pink color and chain. So this part of this figure was already there. Now, using in vitro cell line study, we have studied the signaling pathway. So we used monocytes. Monocytes, when stimulated with PMA, forward ester, it is converted into activated macrophage. Activated macrophage was treated with oxidized LDL, and it is converted into foam cells. So you can see the THP1 is a monocyte, and then monocytes is converted into macrophage. Macrophage is converted into foam cells. These are all in vitro culture system, and we have shown that CD36 is upregulated in the foam cells, and the PPR gamma was downregulated in the foam cells, but it is upregulated in the macrophage. So it helps in the maintenance and the cells in the inflammatory stage. With many other experiments, we have summarized here that we published the paper in Journal of Proteomics. where it shows that AZGP1 is released from the adipocytes, and with the help of oxidized LDL, it binds to CD36. And if this drives towards the balance pathway, as long as PPR gamma is downregulated. When PPR gamma is upregulated, it is ABCA5 is upregulated, and the RCT is balanced. Reverse cholesterol transport is maintained. So the cells are in the homeostatic stage. But if this pathway is driven towards the people, when PPR gamma is downregulated, then in chronic induction of chronic inflammation occurs, along with PGL or RP2, which we have identified in the mass spec, and also we have validated by using proteomics and the Western blot analysis in the human plasma samples. It helps. We have seen that it is increased, so it helps in the prolonged inflammation. Also, the C17 over 57 also have shown that downregulated. So all these factors which have identified in the proteins which are identified in the blood samples in the patient, and compared with the healthy control, they demonstrated that the system is driven towards the chronic inflammatory stage, and it is also towards the acute coronary syndrome. So inhibition of PPR gamma through CD36 and AZGP1. So this is all driven by AZGP1. So this is a novel protein we identified, and we have found that this is published in the Journal of Proteomics last year during the lockdown. So then our next question is, we have found in that ABCA5 was upregulated in the plasma, in the orbital mass spec analysis in the patient samples. So question is why? So we have utilized cell culture system, animal model, knockout mice, and the human patient. So this is the last part of my topic. So this gentleman, Alipto Gorai, he developed this in vitro system where he has developed the cholesterol loading and unloading system by using the macrophage. But he has shown that if you treat the cells with the macrophage with the cholesterol, the physiological level of cholesterol in vitro culture system, and with different duration of time, you can see that relative mRNA expression of ABC1, the primary cholesterol transporter, is increased with the duration of cholesterol loading. So you can remember that ABC1 is the primary transporter. ABC1 is increased in response to cholesterol loading. But ABCA5, first time we have reported using mass spec analysis that ABCA5 was upregulated in the MI subjects, and also MI subjects having high cholesterol level. So all MI subjects may not have high cholesterol level. Those subjects, those patients having high cholesterol level, we have found that ABCA5 was upregulated. So the question was, why ABCA5 was upregulated? To answer that question, we developed this model. Olito developed this model, and he has shown that with the duration of cholesterol loading, the ABCA1 is upregulated. When cholesterol was unloaded using HDL, in the in vivo system, HDL helps, high density lipoprotein helps in the unloading of cholesterol into the liver hepatocytes. So that is the normal process for reverse cholesterol transport. So when if unload the cholesterol from the cell, this cholesterol, the ABC1 is downregulated. Same thing happens for ABCA5. So levels of ABCA1, ABCA5 mRNA in raw 264 point cells after free cholesterol loading and subsequent loading using HDL. So raw 264 point cells is the macrophage cell. So this shows that ABCA5 is a sterol responsive gene. ABCA1 is a sterol responsive gene is already known. We have shown here that ABCA5 is also sterol responsive gene. So now the same condition, similar situation. So we have increased the cholesterol level. What Olipto has treated the cells with increasing concentration of cholesterol. 100 micromolar cholesterol is the physiological level of cholesterol, and 150 micromolar cholesterol in the in vitro system is the high cholesterol load. So with the high cholesterol loading, very intriguingly, that ABCA1 is downregulated. But ABC1 should be upregulated because ABC1 is the primary cholesterol transporter. But you can see that ABCA1 is downregulated at the high cholesterol level. But ABCA5 is upregulated at the same time. So when ABCA1 is downregulated, ABCA5 is upregulated, which is validated using Western blot and also real-time PCR. Now, he has again conducted the MSMS analysis using orbital mass spec, after treating the cells with different concentration of cholesterol, the macrophage cells, the raw cells, and he has identified the ABCA5 high ABCA5 the level, the peak, because of the 100 micromolar cholesterol, and it also he has identified that fatty acid beta oxidation pathway is downregulated in the because of 150 micromolar level of cholesterol, but as long as the loading was 100 micromolar cholesterol, the fatty acid oxidation pathway was upregulated, and also related signaling cascade also was altered. So the question was, what is the effect of cholesterol overloading at the molecular level? So what is the mechanism? Why cholesterol increases the ABC1 level, and why cholesterol, how cholesterol reduces increase ABCA5 level? So we have identified the transcription factors LXR alpha, LXR beta, and PPAR gamma. LXR alpha, LXR beta are known, or PPAR gamma are known as the transcription factors involved in the expression of ABCO1. So he has shown that with the increasing concentration of because of 100 micromolar cholesterol, LXR alpha was upregulated, but because of 100 due to 50 micromolar cholesterol, LXR alpha is downregulated. So that is interesting. Since LXR alpha is downregulated, beta is downregulated, and PPAR gamma is also downregulated compared to 100 micromolar cholesterol, ABCO or possibly ABCO1 expression was decreased. So, and very interesting, what is the cell fate? What is happening to the cell? Macrophage cells, raw cells. So raw cells, the apoptotic, the cells become apoptotic. So most of the cells become early apoptosis because of the LXR, because of cholesterol overloading. So if you load the cells with high concentration of cholesterol, cells will be gradually moved toward the late apoptosis, and also as most of the cells will be in the early apoptosis. So the apoptosis cell will be, but apoptosis will be there, necrotic will be less, and most of the cells will go toward the programmed cell death. If you overload the cells with the high cholesterol concentration. Now, question was whether these cells, the ABCO1 and ABCA5 co-localized. Yes, what is the location of the ABCO1 and ABCA5? ABCA1 location is known. ABCA1 is localized in the plasma membrane because it is the membrane, it is the membrane cholesterol transporter. ABCA5 is also localized in the membrane, and you can see they are co-localized. So they are the transporter protein. They are supposed to be in the membrane, and also with the cholesterol, we have shown that it is also localized with body peak cholesterol. ABCO1 and ABCA5 both co-localized with the body peak cholesterol in response to the stimulator. And then we checked, we asked the question whether ABCO1 will be altered because of the transcription stimulation with the small molecule of the LXR1 alpha, LXR1 activator. So GW3965 is well-known LXR activator. Because of the treatment of the cells with GW3965, or if you treat the cells with LXR1 inducer, the ABCA1 is increased, and ABCA, but with the help, but in the presence of cholesterol, ABCA1 was decreased. At the same time, ABCA5 was increased. So this is very interesting. So ABCA1 and ABCA5 has a different, the inverse relationship. The condition when ABCA1 is decreased, the ABCA5 was increased. So that is very interesting. That is an interesting clue towards our story. And the ABCA1 also stimulate, and also the cells were stimulated with the PPAR alpha, PPAR alpha and gamma, general PPAR, PPAR alpha, PPAR gamma inhibitor activator, and it is shown that these are all stimulated with PPAR alpha and PPAR gamma. So this is already known that ABCA1 and ABCA5, ABCA1 is stimulated by PPAR alpha. So this ABCA1 is synthesized by the PPAR alpha signaling cascade. But ABCA5 is not stimulated by PPAR alpha cascade. ABCA5 is stimulated by PPAR gamma cascade activator. So rosiglitazone, troglitazone are the PPAR gamma activator. So if you treat the cells with troglitazone or rosiglitazone, you can see the ABCA1 was in ABCA1 is increased, but ABCA5 was also increased. So because of PPAR gamma activator, the PPAR in the presence of cholesterol, ABCA5 was increased. So ABCA5 is a PPAR family regulated gene with more specificity for PPAR gamma isoform. So So PPR gamma, we first time showed that ABCA5 is stimulated is through expressed through PPR gamma pathway. Now we have also now shown that what if we treat the cells with different lipid modulator of lipid homeostasis? Somehow, if we can increase the cholesterol concentration in the cell, how can we increase the cholesterol concentration in the cell? By treating the cells with atorvastatin. You know, the atorvastatin or statin is the inhibitor of the first, the very key step in the cholesterol synthesis. This so because of this, the cholesterol synthesis is inhibited. So if you inhibit the cholesterol, increase the cholesterol by atorvastatin, so reduce the cholesterol by atorvastatin, ABC1 increased. But if you treat with the cholesterol, then ABC1 is restored. But at the same time, ABCA5 was increased. So if you treat the cells with cholesterol, with the lipid modulator of lipid homeostasis, ABC1 and ABCA5 expressions are altered. But ABC1 and ABCA5 behave differently. If you treat the cells with oxidized LDL, ABCA1 is increased. But if you treat the cell with oxidized LDL plus atorvastatin, so atorvastatin is the inhibitor of HMG-CoA reductase. HMG-CoA reductase is the first step in the cholesterol biosynthesis. If you treat the cells with atorvastatin, cholesterol will be lower. Now, if you treat the cells with oxidized LDL and cholesterol, oxidized LDL, the ABCA1 is increased, ABCA5 increased, but in the presence of atorvastatin, ABCA5 remains at high concentration. So this is validated by Western blotting and the mRNA level, real-time RT-PCR. So ABCA5 expression is increased in all studied cases of ABCA1 downregulation, indicating a compensatory response. First time now we are studying this word. There is a relationship. There is a compensatory relationship. ABCA1, ABCA5 has a compensatory relationship. So whenever ABCA1 is increased, ABCA5 is decreased. So we created this situation. Now what happens to cholesterol efflux? Efflux is the actual function. So if you treat the cells with body cholesterol at the 100 micromolar cholesterol or the increasing cholesterol load, the cholesterol efflux will be increased. That is that is expected. So, but when atorvastatin will be there, the ABC1 mRNA, we have seen that ABC1 mRNA decreased, cholesterol efflux will be inhibited. So, if you treat the cells with atorvastatin, the ABC1 was decreased, and the primary cholesterol transporter is decreased, and the cholesterol efflux was affected. So, cholesterol efflux is decreased. But if you then silence the ABC1, so to establish the cause-effect relationship, we have to silence this protein. So we silence the ABCA5 first. So if you ABCA1, if we silence the ABCA1 and treat with 100 micromolar cholesterol, you can see ABCA5 is increased. See, ABCA1 was silenced in the cells are knocked down using the siRNA. ABCA1 was decreased in the presence of 100 micromolar cholesterol. ABCA5 was increased. So when ABCA1 was decreased, ABCA5 increased. So we have created this situation in the in vitro condition. Same thing happens to when ABCA5 was down-regulated, locked down. So when ABCA1 is knocked down using siRNA, and also in the presence of 100 micromolar cholesterol, ABCA5 was knocked down, ABCA1 was increased. ABCA5 is there, ABCA1 was decreased. In the high cholesterol concentration, 100 micromolar cholesterol concentration, in the absence of ABCA1, ABCA5, ABCA1 is not increased. That is very critical. In the presence of 100 micromolar cholesterol, ABCA5 is decreased, ABCA1 increased. So ABCA1 can compensate loss of ABCA5 function at the lower cholesterol physiological level, because it's the primary transporter of cholesterol. But in the higher cholesterol concentration, ABCA1 cannot manage ABCA5 function. And what is happening in the cellular status? This is very critical information. When ABCA5 was downregulated, see, most of the cells are going to late apoptosis. So number of cells are going to necrotic, number of cells are increasing towards the late apoptosis when the cells are treated with high 150 micromolar cholesterol when ABCA5 was downregulated. So that is actually the situation in the plaque, in the advanced plaque. In the advanced plaque, the atherosclerotic plaque core necrotic plaque area is increased, cells become more apoptotic, cells become more necrotic also. So it is actually mimicking the advanced stage when the IBCA5 is also decreased. So now the question, so we have all they established this cholesterol transporting mechanism in the in vitro, in the in vivo also, in the in vitro. Now let's see in the in vivo condition. For in vivo, we have used the two kinds of knockout mice. One is the apo knockout mice model. It is the atherosclerotic model. This is popularly known model for atherosclerotic study. And also we have used another model, which is alpha knockout mice model, which is hyperlipidemia model. Let's see what is happening. So if you feed these animals with high fat diet, and you will see that there is a huge atherosclerotic plaque, and this area is the lumen is decreased, so atherosclerotic plaque is increased. So you can see this very advanced stage of plaque formation after 2 months of feeding with the high cholesterol diet in the apoinocord mice or in the PPR alpha knockout mice. You can see the normal lumen in the aorta in the longitudinal sections after 2 months of the normal diet for feeding, and after 2 months of feeding with high cholesterol diet, you can see the necrotic core area, almost plaque is going to be ruptured. This is in the animal model. So in this condition, I'm not going to the detailed data, just you can see that whenever high cholesterol diet is there, so mice fed with high cholesterol diet for different months, different duration, 45 days, 60 days, and 90 days, in the PPR alpha knockout mice, you can see that ABCA5 was increased, ABCA1 was decreased, ABCA5 was increased, and ABCA1 was decreased in the mice after 90 days of feeding. What is PPR alpha knockout mice? I forgot to mention you. PPR alpha knockout mice, because of a PPR alpha is knocked down, PPR alpha is defective, the fatty acid oxidation is hampered. There is no fatty acid oxidation. Because of lack of fatty acid oxidation, cholesterol level is very high in these mice without being, and if you feed these mice with high cholesterol diet, high fat diet, the high cholesterol, high fat diet, the cholesterol level is very high. So we have created the condition that, okay, let's create the situation where the mice is having high cholesterol load. So if you have high cholesterol, mice with high cholesterol, you can see the ABCA5, the secondary transporter is increased, the primary transporter is decreased. This transporter, how do we analyze, find? How did we find this? We found this using orbital analysis in the patient sample first time. Now we have validated in the in vitro system, we have validated in the animal model also. Now next see the another mice model. This is the mice with apo knockout mice. In this mice, because of the apo gene is knocked down, this mice has a huge problem in the cholesterol transport. So the cholesterol level is high, and if you feed this mice with a high cholesterol diet for 30 days, 45 days, and 60 days, you can see that ABCA1 is decreased, ABCA5 was increased. ABCA1 has decreased, ABCA5 was decreased. So whenever ABCA1 is decreased, ABCA5 was increased. So we have established in these mice. So these mice develops atherosclerotic plaque if you feed these mice with high cholesterol diet. So cyclodine levels of ABCA1, ABCA5 indicates cholesterol load. In the other way also, if we check this ABCA5 expression, ABCA1 expression in the blood, if you can indirectly say the cholesterol load in the blood. These are all done using the Western blot analysis and also sometimes ELISA. So now the last validation in the human model, human validation. So we have also validated the animal model. Now let's see the human data. We have started with human data, of course, in the orbital analysis. You can see, so we have collected this information from the so-called normal people, not the patient. So we have collected age, weight, smoking, and the echocardiographic parameters, LVEF, ejection fraction. Now they are relatively fine, fair. Aortic root diameter, glucose fasting, cholesterol level, HDL, LDL, triglyceride, VLDL, etc. So we can see that, and what Olipto did, Olipto has grouped this into 4 classes based on the plasma cholesterol. Plasma cholesterol may have low cholesterol, may have high cholesterol. But low cholesterol may have low HDL or high HDL. So group 1, group 3, and high cholesterol may have low HDL and high HDL. So high cholesterol, having low cholesterol, having high HDL is good. Low cholesterol, low HDL is not good. Low high, so basically high, still it is cholesterol is low HDL means still cholesterol is high here, though is low cholesterol, but high cholesterol patient, they have subjects, they're not patients, they're subjects, so-called normal healthy subjects having risk factors for atherosclerosis. This high cholesterol group has low HDL. That means cholesterol is very high because this cholesterol is not cannot be managed by the reverse cholesterol transport. HDL is the main player and also may have high HDL. Now let's see. So I'm not going to all the detail what is happening for ABCA1 and ABCA5 level. That is our interest here. ABCA1 and level is decreased, but ABCA5 is increased when in the group compared with high cholesterol level. So whatever we have found that the inverse relationship between ABCA1 and ABCA5 with respect to human plasma cholesterol levels of the high HDL, high triglyceride group is confirmed to be consistent with our findings in mice model. So wherever high cholesterol level, ABCA1 is the primary cholesterol transporter is decreased and the secondary transporter is increased. Then we ask the questions, are they structurally different? Why ABCA1 and ABCA5 is being differently? Then we took the help of the computational study and the molecular dynamics simulation. Using molecular dynamics simulation, we have shown that basically they have this similar topology. Because there is a high homology between ABCA1 and ABCA5. The topology is very similar. The V7 in transmembrane domain is very similar, but it revealed a very interesting information using the massive molecular dynamics stimulation by these gentlemen, Sandeepan Chakraborty and the Dhiman Jana in Indian Association for the Cultivation of Science, ICS. Very interesting information revealed here. Normally, the ABCA1 is a primary transporter. This is a normal cholesterol transporter. So, cholesterol reflux actually happens through this cholesterol transport domain, the channel through active transport. But secondary ABCA transporter doesn't look like ABCA transporter, the cholesterol transporter. This secondary transporter is open, behave like cholesterol transporter. It can transport cholesterol when there is excess cholesterol is there, which is actually we found this in the patient sample, in the in vitro system, in the in vivo system. Now, using molecular dynamics simulation, these people established that yes, that is correct. When this transporter is exposed to high cholesterol load, then only it is opened and cholesterol is transported. So there is a cholesterol efflux happens. So under physiological doses of cholesterol, ABCA1 is the primary efflux mediator by an energy-dependent process, while ABCA5 acts as a secondary transporter. Under excess cholesterol, downregulation of lipid metabolism results in reduced core role of energy-dependent ABCA1 for a cholesterol efflux, thereby promoting ABCA5 as the primary mediator, whereby it adapts to a gradient-based efflux mechanism by conformational change. So I say adaptation. So because being the geologist we find this adaptation. So system is trying to adapt. So adaptive mechanism from when the cholesterol level is high, but the cholesterol transporter is low. So system is trying to manage the cholesterol level and trying to maintain the cholesterol homeostasis using another transporter, which we first time discovered. And this is published in Journal of Molecular Biology in the lockdown period 2020. Also, so this is the last slide, almost last slide. I have the acknowledgement slide. And also I am to inform that another paper is very interesting paper is published in MCV in January 2021 this year. And this is also cover page. So if you open this journal, MCV 2021 January issue, you will see the cover page having mitochondrial structure, beautiful mitochondrial picture in the hypertrophic cardiomyocytes because of the knockdown of a very interesting protein, which is regulatory position alpha cascade, which I have not spoken here. That is a very purely in vitro study and also animal model study, no relationship with human samples. So this is very basic fundamental study where the Dibbanti has done. It's published in MCP as a cover page article. I'm very happy to share this information with you. So if you're interested, you can see this in our journal paper. I have not discussed very detail about the molecular dynamics because I am not expert on it. If you are interested, if you can see this journal paper and find very interesting information shared by Sandeepan Chakraborty and Bhimanjana, they are expert in the field. My acknowledgement goes to the Jagdisha Prasad and Mahesh Kulkarni. Mahesh Kulkarni is a scientist in the NCL Pune, and Jagdisha was student for SWOT analysis, sequential window acquisition for all theoretical mass spectra. There is a powerful mass spectrometry for quantitative analysis. We also acknowledge clinicians, Prakash Prachanda Mondol as cardiologist in Apollo Clinical Hospitals, Dr. Kanwar Khawar and Siddiqui, cardiologist, Ruby General Hospitals, and the structural studies were done by Sandeepan Nandanjana in ISCS. Sandeepan has left ISCS very recently. Most of the studies I discussed here were done by Obabrito. He is now in USA. Olipto did the ABC1, ABC5 work. Devasmita is doing another interesting study in the inflammatory pathway. We are sending this manuscript, and this led these students. The Praditushti is also continuing this work in the human samples, using human samples and also in vitro analysis. These are the people using the clinicals work. And these are the people mostly they do the hypertrophy work in the in vitro cell culture system. Thank you very much for your patient hearing. Thank you. Now I will unshare this slide so that you can talk and ask questions.
Yes, sir. Thank you for your wonderful lecture, sir. We have some questions, and can we just have that questions?
Yeah. I can
Okay.
change this the laptop because here camera is not working. You can ask the question meanwhile.
Okay, sir. Our first question is why MS is preferred and why MS is preferred. We can also rely on microarray assay, SAGE, and CAGE for differential analysis.
Yeah, that is a different kind of question. You have asked the question, is fine, but microarray for the gene expression analysis. MS mass spectrometry is for the protein because we are interested for protein, identification of protein. Protein is the candidate which actually molecule function, and the another purpose was to find out the candidate protein which would be used for diagnostic marker. Our final goal is to identify several proteins or the signature proteins which could be used to predict the atherosclerotic burden or the occurrence of acute coronary syndrome in the human patient. That is why we targeted protein, and MS is the preferred technology for mass protein identification.
Okay, sir. Our next question is every individual has different genomic composition, so how it would be validated until and unless large sample size is taken? If taken, so there are different environmental factors. Please explain.
Every individual has different genomic composition to show it would be validated until there are a lot of heterogeneities there. The validation in the large sample size means say thousands or 10,000 or 1 lakh patient is necessary. That is for any understand finding diagnostic marker. But here we are just trying to understand the pathophysiology. For that, 150 samples are enough to understand the system. So this is a basic understanding of the mechanism, not targeting any diagnostic marker. For developing diagnostic marker, yes, large number of sample size is necessary because of the heterogeneity, and that we have not done here. And also multiple cohort studies are necessary. I know it.
Okay, sir. Our next question is there any conformational change in ABCA1 and ABCA5 transmembrane domain, if which specific amino acids are responsible for switching on ABCA1, ABCA5?
Yeah. So this is very nice question. It is this detailed study is done, but there is a conformational change. That I showed the last figure. I have not discussed in detail. As I said, that these people are expert; they have done it. You can find out the paper. Specific amino acids are also indicated there. If you are interested, you just find out the paper. There is exhaustive list, and but this is a computational work, the molecular dynamics simulation. So don't trust too much of the about the amino acid. Those amino acids are changed, but this is an indicative. Uh, for that, to understand the specific amino acid very confirmably, to confirm that, you have to do structural biology and the other studies. That is not done. Based on the molecular dynamic solution, there are specific amino acids. So please refer to the paper.
Okay, sir. Our next question is, can SNP studies of ABCA1 and ABCA5 predict susceptibility to CAD?
See, ABCA1, ABCA5 level is enough to predict susceptibility to CAD. So why you'll go for SNP? Because if you find protein and which can be easily demonstrated by using the ELISA, why you'll go for SNP study? Because for SNP you have to again validate, you have to find out what is the frequency, how many sample it happens. It may not have any relationship. SNP is the completely different approach. SNP may not be used for predicting CAD since you have a protein. So yes, ABCO1, you have indicated one very interesting thing, susceptibility to CAD. That is our final goal. So the new idea is not the ABCO1, ABCA5. We have identified a couple of proteins. The many people are trying to find out some of the signature proteins. So the say 10 proteins are altered in the at the time of heart attack or before heart attack. So these 10 proteins up and down regulation, with the help of AI, we can find out biomarker, we can develop a diagnostic tool to predict CAD. That is the final goal. For that, we have to validate in large number of samples and different cohorts. So you don't so therefore, answer to your question, the ABC1, ABC5 along with other proteins will be enough to predict the susceptibility of CAD. You don't need SNP.
Okay, sir. And now the last question is from Akashlina only. In high cholesterol mice model, what kind of dietary fat and in what ratio has been used?
That is discussed in detailed methodology. This is that the high cholesterol that is uh, manufactured, purchased, the very standard diet purchased from abroad. High cholesterol diet is high. The compositions are there. So I don't remember the composition. These are mostly purchased from very renowned company, well accepted high cholesterol diet. Again, I'll suggest you to, if you want to see the actual composition, you just go through the paper.
Okay, sir. These are all questions, and thank you, sir, for explaining and talking to in our platform. And now I would request Professor Ena Rai Banerjee, our mentor and convener, to give the ending note.
Yeah,
Over to
so
you, ma'am.
I have found the paper that 25% carbohydrate protein, 58% carbohydrate, 17% fat, is the high cholesterol diet purchased from research diets from America, so I think so abroad.
Oh, that's excellent.
That is the high
I hope
cholesterol.
Akashlina is listening. Thank you, Professor
Yes, sir.
Bandyopadhyay, Arunda. Our gratitude to you for speaking on this platform, and congratulations for those two wonderful publications, especially the one that made the cover of MCB. Congratulations for that, and excellent
Thank you,
work
thank you.
that you've been doing. So best wishes for that. I wish I could see you, but your video is probably not on or something the matter
Yeah,
with your
yeah, the
system.
camera is not taking. Yeah, this
Yeah.
is the issue.
So I'll just imagine that you're there.
Yeah,
As is the convention,
yeah,
the students will probably have more questions, but
yeah.
I will not ask any more scientific questions. Probably that discussion we could take offline. But as is the convention, we would like to end the talk, since you are an alumnus of zoology. zoology, we would end the talk by requesting you for a message for the students of zoology and maybe share your journey from zoology to where you are today.
Yeah, so that's very interesting. Zoology is the best subject in the world. If you cannot, you do if you don't trust me, just read the book Richard Dawkins. Richard Dawkins very clearly said and explained why zoology is the best subject in the world. So I have a couple of series of books of Richard Dawkins. So when I give popular lecture, I quote from him, and I got many ideas. So every zoologist should read Richard Dawkins. You know Richard Dawkins, the professor in Oxford. Now he may be 80 plus years old. He has published several books, including so at least 15, 16 books, and I have many of his books. So he has very nicely said that why this zoology is the best subject, and he has actually I regret sometimes that why I couldn't get exposed to Richard Dawkins' book when I studied zoology. I would have moved to Oxford to to do postdoc under him. So this is a fantastic person, and he explained very nicely. So the adaptive complexity, actually, I use the term in many papers, in our technical papers also. So basically, adaptive complexity. This is the adaptive complexity, example of adaptive complexity. Adaptation requires a lot of time. That is the adaptation evolutionary time scale. But I can see adaptation takes place inside our physiological system. So this ABCA1, ABCA5 is the adaptive complexity, and that helps the ABCA5 to increase. There's a fantastic information. I actually got this idea after reading the Richard Dawkins. So all the zoology students must read Richard Dawkins, and finally the extended phenotype, the book Extended Phenotype. Many of you know what is phenotype, but many of you don't know what is extended phenotype. If you want to know extended phenotype, you just read this book. In another occasion, I'll talk about that. Thank you very much.
Thank you, Orunda, and we do hope you will come back. And thank you to all the viewers with us. Thank you, Shakti,
Thank you.
for moderating the show, and we hope to have many more stimulating talks like tonight.
Thank you. Thank you very much.
Thank you, ma'am. Thank you, sir. Now to our audience, you can like and share these videos with your friends and colleagues, and subscribe this channel to watch other videos which are archived in our channel. And our next webinar series are on 1st and 2nd May 2021, and hope you will enjoy those talks also, and please join those talks and listen to those. And till then, stay safe, and thank you.